Hello and welcome to this video lesson on proteins with a main focus on enzymes. Enzymes are a type of proteins, but after a short introduction into proteins, we will look in great detail about how enzymes are really important, their structure and function in all living things. Okay, so in this lesson we're going to be looking at what are proteins and where they are found in our body and other living things including plants, enzymes, a type of proteins, and understand why they are known as biological catalysts and their importance, structure and function. We'll also look at in how enzymes, different types of enzymes, play a huge part in our digestive system in digesting and breaking down the food we eat. And we'll also look at different types of enzymes, how they break down food into smaller molecules, which is known as chemical digestion. And lastly, we'll look at how enzymes are affected by pH and temperature. So, what are proteins and where are they found in our body, in humans? We can say 20% of our body mass is made of proteins. So that means it is pretty much a building block of nearly all living things. Proteins are very large molecules found in living things. They are made up of mainly these elements: carbon, hydrogen, oxygen, nitrogen, and sulfur. These elements actually make a smaller compound known as amino acids. We will look at this a little bit later, how amino acids join together and make a huge molecule known as proteins. So amino acids are the smaller molecules which form the larger molecule protein. So we'll look at that a little bit later, how that works. So where are these protein found in human body? One good example is the muscle tissue. Proteins form a structural part of muscle tissue. What we mean by that is it forms parts of the muscle tissue. The main parts of the muscle tissue is made up of proteins. Now, if you look closely at muscle tissue, it's made up of these fibers, muscle fibers, which have the special property to be able to contract and relax. As you know, which is the main property of muscles, being able to contract and relax. So it is an integral part of muscle tissue. So it is enough if you are able to say muscle tissues or proteins form structural component of some tissues, for example muscle tissues. Okay, for examination purposes, that's enough if you're able to say that. What I've shown is to understand where exactly these tissues, these proteins are, and how they are part of the muscle tissue. Hormones, one of the most important chemicals which carry out specific functions in our body, such as insulin, for example, they're also made up of proteins. Okay, so this is a good example of a protein which controls blood sugar level. As you know, insulin is such an important chemical that's made in pancreas. It is an example of a hormone. There are many other hormones made, and all these hormones are made up of proteins. We also have antibodies made by white blood cells. As you know, these are the chemicals which fight diseases. That they also contain, they're made up of proteins as well. They fight bacteria and other microorganisms. In fact, you can call them as pathogens. Okay, so they attach to these microorganisms and destroy them. So antibodies are good examples of proteins. Another example of protein. Most important of all, and that's most of this video lesson is going to be centered around this, are enzymes. Enzymes are proteins basically, and they are also dubbed as biological catalysts. Okay, because that's what they do. They speed up chemical reactions that happen in our body. For example, breaking down carbohydrates, which is our main source of energy, into glucose. So that sort of thing is done by enzymes, which are proteins. Okay, and many other cell parts are made up of proteins. Okay, these are the main examples. If you want to know a little bit more about antibodies, that's what I'm going to show now. Okay, so here you have a white blood cell producing antibodies. When the body recognizes there is an microorganism which is harmful to our body, it starts to produce these antibodies. A special type of white blood cell start to produce these antibodies. Now these are protein. It's a type of protein, and it's important to know and be able to say as an example of protein in our body. Antibodies made up of protein, and what they do is they attach to these harmful microorganisms in special sites, and they have this particular shape. And the shapes for these proteins are really, really, really important, because their function depends on this particular shape. As you can see, it's roughly Y-shaped, and it helps them to attach to these specific sites on the bacteria or other harmful microorganisms that's come inside our body. What you see here is antibody, which is got that Y shape, and you can see it is a huge molecule made up of like thousands of amino acid units. Okay, so this is an example of a protein. So we'll see how a protein molecule has this particular shape. You can see there's quite a lot of folding in this particular specific shape that it's got. So let's have a look at what protein is made up of. As I've just said, proteins can be made up of anywhere between a few hundred to thousands of these amino acid molecules. Now these molecules are smaller molecules, and when they are joined together in a specific way, chemical bonds are formed between them, and they join up like this, and this keeps going. It's only part of the protein molecule shown here, a very small section shown here. You can see the protein molecules linked together in a particular order, and this sequence determine what protein is being made, and the final protein is actually heavily folded into a specific shape, which determines what type of protein it is and how it works. There are just 20 naturally occurring amino acids that are found in living things like plants and animals, but there are many different types of protein, only because these amino acids can be joined up in different sequences. Now, depending on the sequences you get and the size of them, you get different types of protein. For instance, a good example is, you have alphabets A to Z. That's a good analogy. You can have millions of words by using different sequence of alphabets from A to Z. In a similar way, you can have different types of proteins, thousands of types of proteins, by just having different sequence of these amino acids bonded together to form the larger molecule protein. Plants also have a protein, and protein is really important for plants and for their growth and repair of damaged parts. So just to name a few, chloroplasts, most of the cell parts contain protein. For example, chloroplast is made up of protein, not just protein, but mostly protein. Mitochondria, again, protein is involved there. So mitochondria is mainly made up of protein. Enzymes are just protein, and to make these proteins, for the plant to make this protein, it needs this element from soil, nitrogen, and it's important for its growth as well. So if plants don't make protein, it doesn't grow properly. If it doesn't get enough nitrogen, its growth is affected. So for a plant to grow healthily, it needs to make a lot of protein, and it needs nitrogen from the soil. Plants do store a lot of the protein that it's made in the seeds for the plant to germinate, for the seed to germinate, and for the young growing plant, because it's so important for the growth of the new plant that is its offspring in its early days. So most of the protein the plant makes gets stored in the seeds and for instance nuts and lentils. As we already know, enzymes are proteins, but they are made inside the cells. So the lining, as an example, the lining of the small intestine, it makes enzymes, different types of enzymes, but some of these enzymes work outside of the cells. For example, these enzymes actually are released into the food passage, into the food, where it actually breaks down the food chemically into smaller molecules so it can be absorbed into the bloodstream. So it's really important to realize that some enzymes actually are released, come out of the cells and work outside of the cells. In this part, we are going to be looking at how different types of enzymes act on the food, different types of nutrients, to break them down into soluble, smaller molecules, and that can be absorbed into the bloodstream, making them more useful. Okay, so it's about chemical digestion. How these enzymes play a crucial part in that chemical digestion process is what we're going to look at now. To start with, we are going to start with saliva, which is in the mouth. So as you chew the food in the mouth, this enzyme is secreted by the saliva. Saliva contains amylase. It's a type of enzyme which breaks down carbohydrate. Don't worry about remembering all this. We'll see a summary in a minute. But for now, we're just looking at what enzymes are produced where and what do they break down. So amylase break down carbohydrate. Carbohydrates are broken down, for example, starch present in potato, pasta, rice, and all these bread. They are broken down in the mouth to some extent, not all of it, by the action of this enzyme amylase. Okay, how it works, don't worry, we'll look at this in a little while. Then food arrives here in the stomach. Now stomach has acidic condition because it has hydrochloric acid made by the walls of the stomach. So hydrochloric acid is made here, and in this acidic condition these enzymes really work well. Okay, so it's important to realize by the time the food arrives in the stomach, acid is added to it, a very strong acid which lowers the pH to around about 2. Okay, so in the stomach you have this enzyme made by the walls of the stomach, protease, which breaks down protein. Then the food starts to move from the stomach down to the small intestine, when, as you can see here, this part, pancreas, adds all the 3 enzymes to the food. Amylase, you can see that's joined, pancreas is joined to amylase, which is that part there, pancreas makes that enzyme amylase. You already know salivary gland also makes amylase, but now you know pancreas also makes amylase, which breaks down carbohydrates. Okay, pancreas, as you can see, another line joins up here. Pancreas makes the enzyme protease, which you know it breaks down protein. Pancreas also makes lipase. So pancreas is such an important organ in the digestive system, as it makes all the 3 main types of enzymes needed for the chemical digestion. So it makes amylase, protease, and lipase. Okay, and it adds it to the food, which breaks down carbohydrates, proteins, and fats into smaller soluble molecules. Okay, so as the food travels into the small intestine, the food is slightly alkaline here in this part because something is added from the liver. Liver makes bile, which is stored in the gallbladder, and it's added to the food, which has got 2 functions, but mainly we'll look at that in a few minutes, but mainly it makes the food alkaline. So food arrives here in the small intestine. In the small intestine, again, more enzymes are made by the cells in the walls of the small intestine. So let's have a look at what enzymes are made there and added to the food. Protease, as you can see that line joining, which breaks down protein. You have also amylase made in the small intestine, Small intestine also makes lipase. So basically, as it goes past the stomach, nearly all the enzymes are added: amylase, protease, lipase by pancreas, as well as the small intestine. So food gets broken down into soluble smaller molecules, and you know which part makes which enzymes from this diagram here. Okay, it's important that you know exactly what enzymes are made where, so make sure that you know all of this before you go further. So here is a summary of the enzymes and where they are produced in the digestive system. Make sure you remember these. We already know enzymes break down bigger molecules into smaller molecules in the chemical digestion process. So here is a bigger molecule, starch, and it's broken down into glucose. And it's really important to realize and be able to recognize the three main types of enzymes we've just seen in the digestive system. So what we have here is a bigger molecule broken down into smaller molecules. Now, we call this as chemical digestion. Now, when it's broken down into smaller molecules, it's able to diffuse through the small intestine, the walls of the small intestine, as you know, through the villi into the blood, and only then our body can use this. Body cannot use starch. It has to be digested into glucose, and only then it can go into the bloodstream to be used by the body for energy. Okay, and that's done by this important enzyme carbohydrates. And you know from the previous diagram, it's made in the saliva. So actual chemical digestion starts from the mouth, and by the chewing process it actually increases the surface area of the food, where more enzyme comes into contact with food. It breaks down the starch into glucose. So it's really important to remember the type of enzyme which acts on starch. Also notice that all the enzymes end with ASE: carbohydrates, amylase. These are amylase is a specific name. Carbohydrates is a general name which is given to any enzyme which acts on carbohydrates. Here is another enzyme, protease, which breaks down proteins, which is a larger molecule, into smaller molecules which are now soluble in water. Now remember, as it is, proteins are not useful. When we eat anything that contains proteins such as meat, it has to be digested in our digestive system, broken down by these chemicals we know as enzymes. In this example, in this what we see here is protein being broken down into amino acids by a specific enzyme called protease. In a similar way, fats are broken down by an enzyme, lipase, into glycerol and fatty acids. Now these are soluble in water. These can go into the bloodstream, which again can be made synthesized back into fats to be used for insulation and for many other purposes such as making cell membranes. Okay, so all you need to know is what enzyme is involved, lipase, and it breaks down the bigger molecule, the name of the bigger molecule, fats, and it's broken down into the products, glycerol and the fatty acids, and know that these are the smaller molecules. You should also be able to write word equation for any of these three types of enzymes and the main bigger molecule and the products that are made by breaking down the bigger molecule. Here are the three word equations that you need to know, and you must use the correct. Then you need to know the name of the enzyme which is breaking down a particular bigger molecule, and you should be able to write word equations in this format. It is important to understand the action of bile in the process of digestion, as it greatly helps breakdown of food, but in a different way. So enzymes play crucial parts in breaking down bigger molecules to smaller molecules, but bile plays another part which aids the process of digestion. Okay, so where is it made? First of all, it's made in the liver. So this part here is liver. It's made there, and the bile, which is a liquid, is stored in the gallbladder. As you can see here, the greenish, the green colored part here, which is inside the liver. Okay, so bile is stored there. It's a liquid, and what it does is when it's added to the small intestine, as the food comes from the stomach into the small intestine, it is very acidic. Now, the enzymes in the small intestine works best in alkaline condition. The enzymes in the stomach works best in acid condition. Okay, so you have the bile added to the small intestine, which neutralizes the acid in the food, okay, and makes it even alkaline. So the enzymes here works best in that condition. That's one main job of bile, to neutralize the food and make it alkaline, makes it suitable for the enzymes in the provides best condition for the enzymes in the small intestine to work. Okay, it also has another important job which is called emulsification of fat. So it emulsifies fat, which means if you consider fat as a big droplet, such as this, so if this is a big chunk of fat, okay, and it is actually emulsified, which means it's made into tiny droplets of fat, like so. Okay, so fat, but now it's droplets. Okay, so what's the purpose of that? Now, when this happens, the surface area hugely increases. So the advantage of this is the surface area increasing. Okay, and it's really important in biology, as you know. Whenever surface area increases, it makes the action of the enzymes, which is lipase in this case, it can act on these droplets much more efficiently than on the bigger chunk because it's got more surface area. So enzymes can actually attack more molecules of fat to break them down much more faster. Okay, so that is another main job of bile, emulsification, and this process of breaking down bigger chunk of fat. It's not fat molecule, it is a lump of fat broken down into droplets of fat, which is a small pieces of fat, which now during that process it's increased the surface area. Okay, so it's really important to realize that the process of emulsification by bile increased the surface area of the fat that's in the food by breaking down slightly bigger lumps of fat into tiny droplets of fat. Let's have a quick look at how enzymes actually work. Now, as I've already said, enzymes have specific shape in which it's got notches like this, which is also got certain shape. Now they work on other molecules to break them down. So in this case, we are looking at how digestion happens. So let's imagine this is an enzyme which does the digestion, such as carbohydrates, and we can imagine this is a bigger molecule, and you can see how the enzyme shape is so that this molecule can fit into this place called active site. So what we have here is enzyme, and this molecule is called the substrate, which is the bigger molecule that gets broken down. Okay, and this place is called the active site. You don't need to know all these detail, but it just helps to understand how the enzymes work. Okay, so all of this you may not need to be able to describe, but it helps to understand, so you can appreciate the shape, the importance of the shape of the active site as well as the shape of the enzyme as a protein. Okay, so what happens here, as you can see, the substrate molecule fits into the enzyme and makes the complex it attaches into the active site, and then it breaks that bond here, so that bond gets broken and you get the smaller molecules. Now this is like our bigger molecules such as starch. You can imagine this as a smaller molecule, the digested molecule now, which is soluble, insoluble, bigger molecule, soluble molecule. So you have the digested products here. What we see here is a graph which shows the effect of temperature on enzymes. It's really important to be able to describe what's happening and explain the shape of the graph here. Okay, as you can see, the rate of the reaction, which is catalyzed by enzymes. We know enzymes are biological catalysts, okay, which means it speeds up chemical reactions inside in the body, such as digestion. Okay, what you see here is as the temperature increases, the rate of the reaction increases. As you see here in this graph, it increases up to that point. So at 40 degree, we call that as optimum temperature for the enzyme to work. Optimum means the most suitable temperature for the enzyme to work. This is true for many enzymes in the human digestive system because at higher temperatures they denature. So we need to know what denature means. And the graph clearly shows this. As the temperature goes higher than 40, the rate of reaction decreases drastically. So by about 50, the rate of reaction is nearly zero or very, very low. And this is because something happens to the enzymes. Its shape changes. When we say denature, what we mean is it actually loses its natural shape. And we also know how important the shape of proteins are for their function. So how do you explain up to this point that rate of the reaction be increasing? That is because at higher temperatures, the molecules move faster, the particles move faster, so the substrate molecules and the enzyme protein molecules, they collide more often. So more collisions happen, which leads to faster reaction here. As you know from rates of reaction, higher the temperature, faster the reaction. But past 40 degrees centigrade, what happens is the rate of the reaction falls down and decreases to nearly zero. We need to see and we need to be able to explain why that is happening. This is what we mean by denaturing. So have a look at this enzyme here. It's not going to be able to fit the substrate into this because it's lost its shape. Now I want you to compare that quickly with the original enzyme shape here. Now as you can see, the substrate can exactly fit into this active site because it's got this shape which allows it to fit neatly into this. And this is why we say shape of the proteins are really important for their function. For the enzymes to work properly, it has to have that shape, okay? But as we know, at higher temperature, what happens is the enzymes denature, and this process is denaturing, which means it loses its natural shape. So what happens is this, and the substrate molecule no longer fits into the active site. Okay, so the enzymes don't work at all. It's destroyed. But the best way to describe that is by using this specific scientific term, denaturing. Enzymes denature at higher temperatures. Enzyme activity is also affected by pH, which means whether they work in acidic condition or alkaline condition. Now, as you can see, this enzyme here, enzyme A, as it's labeled here, it works best around 2 to 3, pH of 2 to 3, which means it works best in acidic conditions. Okay. So this enzyme works in acidic conditions, enzyme A. What about this B? Can you see that it works slightly in alkaline conditions? Okay, so around about 7, just past 7, so alkaline condition. Now if you think about this, which of these enzymes are the enzymes from the stomach? Now you can see in the stomach, because we know stomach has hydrochloric acid, okay, so that its pH would be low, around about this. So it's quite possible that this enzyme A is from the stomach, and this enzyme B is possibly from the small intestine. And as you know, in the small intestine, the food is made alkaline by the addition of bile made in the liver and stored in the gallbladder. You should also be able to say the best optimal pH for each of these enzyme. You can easily draw a line on the peak of this, and you can go down and see where it works the best. You can see this enzyme works the best around about 2.8 pH or 6. This one works at 7.5. Okay. You need to be careful about interpreting graphs. Sometimes in the exams they give graphs like such as this. You just need to look at the labels. So in this case, the labels would be, so here you have temperature on the x-axis and the time to digest starch. So if you're asked which is the best temperature for this particular enzyme, you can see it's around 35, 36. So that's the best temperature for this enzyme here. You can even say about 36 to 37, because that's the point where its digestion has taken place very quickly. It's taken the least amount of time. So when you interpret graphs from experiments on enzymes, you just need to look at the labels very carefully, what it shows, and interpret accordingly. You can also see the time decreases as you increase the temperature up to 37, and then the time to digest increases, and it possibly will increase very high past 50, because at higher temperatures it will just denature. The enzymes will denature. You have reached the end of this lesson. You should now be able to do these 3 things. Now I hope you understand how enzymes work and what proteins are in general, and that they are made up of smaller molecules called amino acids. And I also hope that you understood how enzyme activity is actually affected by factors such as pH and temperature. We've reached the end of this lesson. This has been a bit long and quite a lot of information packed, so hopefully it's been useful to you. Bye now, and I'll hopefully see you in my next video lesson.